DNA replication is a semi-conservative process, which means that one single strand of DNA will become separated, and then a new one will be created to match it. And this occurs in 6 steps. So the first step is that helicase unwinds the DNA and it breaks the A and T and G and C bonds, creating something called the replication fork as it separates the two. Then the SSB proteins will bind to a single strand of DNA to keep the helix open, otherwise they could snap back together very easily. Then primase makes starting points called RNA primers, and then DNA polymerase copies DNA 3' to 5' up those primers while a clamp keeps it attached. RNase H removes the RNA primers and DNA polymerase fills in the gaps. And then 6 DNA ligase attaches the DNA background together, and then it is proofread. Stem cells frequently replicate their DNA because of their vast ability to grow into different things. They self-sustain. They're just way more versatile than regular cells. So that sets them apart from muscle and blood cells that do not replicate themselves. If it goes wrong, it of course can lead to things like cancer and genetic disadvantages and loss of genetic characteristics, but it also can cause evolutionary mutations that are like beneficial, like in the case of Lance Armstrong, that has like hemoglobin that's special. But of course that same evolutionary mutations can harm the creature as well.